Hello everyone. On behalf of the Biochemical Society and Portland Press, may I welcome all to this webinar, which is part of the Biochemistry Focus webinar series. I'm Claire Stanford. I'm Professor of Translational Neuropharmacology at UCL, and it's my pleasure to chair this event. Today's webinar is the first of the year in a series dedicated to early career researchers. The topic of this webinar is developments in neuroscience, and we're delighted to provide this opportunity for Louis, Shaun, Michelle, and Janos to share their exciting research findings with the biosciences community. As with all webinars, you will have a chance to ask questions via text, and I'll explain how to do that in a minute. Other topics in the series of the webinars will include a range of research areas in molecular biosciences, as well as practical sessions to support career development. Of course, we would welcome suggestions for future topics and also speakers to feature in this webinar series, so please do check the website for more details on that and how to submit those ideas. Next slide. Finally, may I draw your attention to the journal Neuronal Signaling, which is part of the Portland Press portfolio and the sponsor for this event. I was really delighted to be asked to be the next editor-in-chief of Neuronal Signaling, which has a distinguished editorial board who are all really well placed to recruit experts for peer review of manuscripts submitted for publication in the journal. Importantly, the journal is listed in PubMed and is also fully open access. The open access fee is waived for researchers whose institutions subscribe to the journal, so do please encourage your institution to subscribe. Also, unlike many journals in the field, pilot illustrations are completely free. We're particularly keen to support early career researchers, so do please consider submitting your next manuscript for publication in neuronal signaling, and do encourage your peers to do the same. Now, just one final point before introducing our first speaker. There will be a question and answer session after all talks are finished, and that will be the final part of the webinar. But please send your questions during the talks. Just type it into the question box, as shown in image on the screen, and state who your question is for, and we will try to answer as many as we can as time allows. So now, on with the talks, which is the key part of the webinar. Our first invited speaker today is Dr. Dwomoh from the University of Glasgow. Now, Louis is a research associate at the University of Glasgow's Centre for Translational Pharmacology. His research focuses on defining novel G-protein coupled receptor targets for modification of neurodegenerative disorders. And today, Louis will discuss the disease-modifying effects of targeting the M1 muscarinic acetylcholine receptor with a novel positive allosteric modulator in prion disease mice. Over to you, Louis.
Right. Thank you very much. I would like to thank the Biochemical Society for this opportunity to share my work, which involves targeting the M1 muscarinic receptor as a therapeutic strategy for Alzheimer's disease. Okay, can you see the screen now?
Yeah.
Yes.
Yes, that's fine.
Sorry for the hiccup. So in most neurodegenerative disorders, including Alzheimer's disease, there is one hallmark, which is cholinergic hypofunction, and this is characterized by selective destruction and loss of cholinergic neurons. So these are neurons that express cholinergic receptors, and these receptors synthesize and secrete the key cholinergic neurotransmitter acetylcholine. So because of this selective loss, there is altered levels of acetylcholine, which leads to disruptions in attention, for example, as well as memory and emotional processing in people with Alzheimer's disease. So one way to do away with this problem is that there's been development of acetylcholinesterase inhibitors. And so these are drugs that block the removal of acetylcholine from the synapse. And in doing so, you end up with whole body upregulation of cholinergic transmission. But the biggest problem with this is the fact that we have acetylcholine in other areas, including the neuromuscular junction. And so this then results in profound off-target dose-related side effects, including nausea, diarrhea, and vomiting. And so this is a big problem for people who take acetylcholinesterase inhibitors. And so that means we need to find a different way of upregulating the cholinergic neurotransmission in Alzheimer's disease, and that is what I do. And so the receptor I work on is the muscarinic receptor, which is a G protein coupled receptor. And there are 5 main subtypes of this receptor. We are interested in the type 1 for the fact that we know it has procognitive function. And so, for example, mice that have the M1 receptor knocked out show significant cognitive deficit, as shown in this fear conditioning experiment. One key aspect of this receptor is that in Alzheimer's disease, the expression of the postsynaptic receptors as well as the signaling of the receptor remains unaltered, so it doesn't change. So we do have a receptor which is not changed in the disease, which will be a very good target if we're able to develop drugs to stimulate this receptor. And so we have an idea, we have a working hypothesis, but we need an animal model. And we do know there are many animal models, but we also have to think about ways to target the receptor. And so the receptor has 2 main binding sites. And the first binding site is the allosteric pocket, which is where acetylcholine, which is the endogenous ligand, will bind. But the problem with this is all the 5 subtypes do have very conserved allosteric pockets, meaning that it is very difficult to develop drugs that will specifically target the M1. And this is exemplified by xanomeline, which was a drug that was made to target the M1 and the M4 subtypes, but ended up having significant off-target side effects and was withdrawn at phase 2 clinical trial level. And so what we do in our group is to target the other site, which is distinct from the allosteric pocket, and it's called the allosteric site. And this site is actually less conserved, so you are able to develop drugs that will bind to the site specifically, for example, for M1. And we know that binding of a drug to the allosteric pocket can increase the affinity and the efficacy of acetylcholine at the allosteric pocket. And these drugs are termed as positive allosteric modulators. And I'll be talking to you about one called the VU846. So there are many Alzheimer's disease mice models that people use in their research. And we use the mice prion neurodegenerative model. And the reason being that this is a terminal neurodegenerative disease that you can actually follow the course of the disease. We've been able to map the timeline. We know what happens at different stages of the disease. And if the mice is left untreated, 12 weeks post-infection, the mice will usually develop confirmatory signs of the disease and die. So having been able to map up what happens in different stages of the disease, we are able to do our experiments based on this. And so we start the administration of our drug or the vehicle at 7 weeks post-infection with a prion protein. We also have another, and so 2 weeks after we've done daily IP injection of the drug, we do the hippocampal-based memory and learning test, which is a fear conditioning test. We also have a cohort where 3 weeks, which is 10 weeks post-infection, we dissect the hippocampus for proteomics and other biochemical analysis. We also have another cohort where we start the drug treatment at week 7, and every day we look for signs and we look for confirmatory signs of the disease. And when we see that, we take the mice and we kill the mice. And we do this in a survival study, which I'll show the results later. So the first thing I want to show is that administration of the VU846 is able to rescue the learning and memory deficit and that we see in the prion mice. So the black bar here shows the prion mice which has not been treated with any drug. So that's a vehicle. And it shows significant learning and memory deficit, which is shown by the time that it spends being immobile which shows a deficit in hippocampal-based learning and memory. But the cohort that has been treated with the VU846, there is a rescue of the learning and memory deficit, as we see here, which is very significant. We've also shown that the mice that have been treated with the VU846 live significantly longer than the mice that have been treated with the vehicle, which is also very important and interesting to observe. So what we've done next is to go down to the biochemical level. We want to see what is happening at the biochemical level, what the prion disease is doing, and if anything that is being changed could be resolved by administration of our VU846. And so we've done mass spectrometry-based proteomics. And we show that when we compare the prion mice to the control, or when we compare the prion-treated mice with the control, in both situations, there is upregulation of key proteins here. But these upregulation are not as intense as we see in the disease state compared to the drug that has been treated. And I will explain this in the next slide. So on this slide, we can show that all the black points I have here are proteins that are significantly either upregulated or downregulated by the prion disease compared to the control. And their corresponding green proteins are then showing that when we treat these mice with a VU846, these proteins that are increased are now being resolved, coming close to the normal, and those that are being reduced are also now being increased or come back to the normal. So a typical example is GFAP here, which we can see almost threefold log two-fold change. But this is halved when we have the mice treated with the VU846. So we are seeing that the prion neurodegeneration is actually changing expression of key proteins in the hippocampus, and these changes are being resolved or being normalized by treatment with the VU846 drug. But we want to see what these proteins are actually doing in the central nervous system. And so if we take the proteins that are significantly reduced in the prion, but then are being normalized or resolved by treatment with a drug, we can show that these proteins are very much involved in neurotransmitter secretion and then synthesis, as well as signaling and synaptic plasticity and memory. So these are processes that are very important for homeostasis in the brain, for learning and memory, and they are being brought down by the disease. But we can show that the treatment with the VU846 is actually trying to resolve this situation. We can also show that most of the proteins are significantly upregulated in the disease state, but are now trying to be resolved by the treatment with a drug. Proteins that are involved in neuroinflammation, for example, which is very important in most neurodegenerative disorders, including Alzheimer's disease. We also see proteins that are involved in proteolysis, which is a response to misfolded proteins, for example. But interestingly, we also see proteins that when you have genetic polymorphisms in their genes, they can either confer risk or protection from Alzheimer's disease. An example is ApoE. So it's quite interesting that we are seeing these same proteins here being expressed or being changed by the prion. And then when we treat these prion mice with our drug, we are seeing that there is a difference, or they are being normalized. And so what I've been able to show today is that we have a very good mice model, which is very good for investigating neurodegenerative disorders because of the timelines that we've been able to map. But it's also very good for drug development and drug discovery. And we also show that the neurodegeneration induced by the prion results in significant changes in key processes in the brain, including inflammation and complement activation, and also disruptions in memory, neurotransmission, and synaptic plasticity. And these abnormalities that we see may underline the cognitive deficits and the shortened lifespan that we see in our neurodegenerative mice. And lastly, the M1 positive allosteric modulator, VU846, modifies the neurodegeneration and improves survival by reversing or normalizing the abnormalities that we see that have been induced by the prion. Thank you for listening.
Really interesting news. Thank you very much for telling us about that really interesting work. I'm sure there'll be lots of questions. We'll pick up those later on. But meanwhile, we move on to our second invited speaker, who is Dr. Shaun Sanders, who is an assistant professor in the Department of Molecular and Cellular Biology at the University of Guelph in Ontario, Canada. Her group works on understanding how neurons use a process known as palmitylation to target proteins to specific subcellular locations. And she's going to describe how that process contributes to normal neuronal function and neuropathology. So over to you, Shaun.
Okay, thank you very much, Claire, for that introduction, and thank you to the Biochemical Society for this opportunity today. So first, as a professor and researcher at the University of Guelph in Ontario, Canada, I would like to acknowledge that the university is situated on the traditional territory and treaty lands of the Mississaugas of the Credit First Nation of the Anishinaabe peoples. So today I'm going to tell you about work I did during my postdoc that was recently published in eLife on population-dependent control of ion channel clustering in neurons. So our nervous system performs many complex functions throughout the day by integrating sensory information to produce an output response. This complex processing involves the electrical impulses firing along circuits and requires the precise control of communication between neurons in the form of action potentials and neurotransmission at synapses. And this in turn requires the precise localization of proteins to specific subcellular locations. The best known of these locations are synaptic contacts, where postsynaptic neurotransmitter receptors are clustered below presynaptic release machinery. And we know a lot about how synapses are organized, and there are two main themes that arise. The first is the presence of scaffold proteins that organize receptors into clusters postsynaptically. The best known of these scaffolds are the family of membrane-associated guanylate kinases, PDZ domain containing scaffolds, including PSD95 and PSD93. So the second theme that arises is that many of these receptors and scaffolds are palmitoylated, and their synaptic clustering is palmitylation dependent. So palmitylation is a post-translational covalent addition of proteins, covalent modification of proteins as cysteine residues with long-chain fatty acids, most commonly palmitate. In the case of cytosolic proteins, palmitylation can serve to tether a protein to a particular membrane. And many transmembrane proteins are also palmitoylated, and palmitylation can serve multiple purposes affecting localization and function, from tilting transmembrane domains in the membrane to regulating interactions with membrane microdomains, other proteins, or other post-translational modifications. So in this way, palmitylation can broadly control multiple aspects of localization and function of ion channels and their scaffolds. So palmitylation is mediated by a large family of DHC domain containing palmitoyl acyltransferases. And while all PATs have the same general domain structure with 4 transmembrane domains and a DHC catalytic domain, some have extended C-terminal tails. And one of these is DHC14, which importantly is the only PAT with a C-terminal type 1 PDZ ligand that is predicted to bind many of the synaptic PDZ domain containing scaffold proteins, including PSD95 and PSD93. So based on this, our initial thought was that DHC14 would be a synaptic regulator. So to identify novel substrates of DHC14, we performed a yeast-2 hybrid screen using the C-terminal tail as bait and a library of hippocampal prey. And we identified one major hit, PSD93. And as I mentioned previously, PSD93 is a synaptic scaffold, but it is also a well-known palmitoylated protein. And so we next asked if DHC14 is a PAT for PSD93 in neurons. So to do so, I developed a DHC14 knockdown system where embryonic rat hippocampal neurons are transduced on DIV9 with lentivirus to express DHC14 shRNA and GFP in 95% of cells. On DIV16, cells are then harvested for biochemistry or fixed for imaging protein distribution. And using this method, I'm able to achieve more than 90% knockdown of DHC14. And indeed, using an assay to isolate palmitoylated proteins from neurons, I see a 60% decrease in palmitylation of PSD93, but no change in palmitylation of another well-palmitoylated neuronal protein, GAP43. And this just indicates that there isn't a global change in palmitylation. So we next asked if loss of PSD93 palmitylation might affect its localization. So I introduced PSC93 to you as a synaptic protein, but I was very aware that palmitoylation of PSC93 is unlikely to be involved in its synaptic localization. And this is based on work done by David Brett's lab in the early 2000s, where mutating the palmitoylation sites in PSC93 has no effect on its punctate synaptic localization, similar to what the wild type form localizes. And so what could the functional role of PSC93 palmitoylation be? And so I realized that PSC93, but not PSC95, a closely related scaffold, is also localized to the axon initial segment here shown with an AIS marker. So could palmitoylation be playing a role in AIS localization? So to just explain the AIS a bit, this is the proximal part of the axon. It has a high density of ion channels and is the site of integration of synaptic input and generation of action potentials. So ion channels cluster at the AIS along with their scaffold proteins, and importantly, many of these ion channels and their scaffolds are also known to be palmitoylated. However, in contrast to how much we know regarding the role of palmitoylation in synaptic protein localization, very little is known regarding the role of palmitoylation of AIS proteins and what transferases control this process. So in our hippocampal neurons, we're also able to see AIS localization marked by anchor and G and indicated here by the arrowheads or contained within this red dotted box, which is blown up in the inset. Interestingly, when we knock down GHC14, we see a reduction of PSC93 targeting to the axon initial segment. And it's just quantified here on this on the right. So what does PSC93 do at the AIS? So PSC93 is involved in clustering of type 1 voltage-gated potassium channels, which importantly also have a type 1 PDZ ligand to bind to PSC93 and are palmitoylated themselves. So we wondered if DHC14 might also palmitylate KV1 channels. And indeed, when we knock down DHC14, we see a significant decrease in palmitylation of KV1.1, 1.2, and 1.4. And these are the three subunits that are found at the AIS and hippocampal neurons. So what about their AIS targeting? And as with PSC93, we see KV1 type potassium channels at the AIS. Here I'm just showing you KV1.4, indicated AISs are indicated by the arrowheads. And when we knock down DHC14, similarly, we also see a reduction in AIS targeting of all three KV1 channels. And again, it's quantified here on the right. So while these findings are very interesting cell biologically, what does it mean functionally for the neuron? And so to address this question, we collaborated with an expert in potassium channel physiology and sent them AAV virus to express the DHC14 shRNA that I optimized in their similar hippocampal culture system. And what they do is patch onto the GFP infected cells, which also receive the shRNA, and inject increasing amounts of current into the cell and measure action potential firing. And they saw an increase in action potential firing after loss of DHC14, as well as a significant delay in or decrease in the latency to the first action potential. So these data are very consistent with loss of KV1 channels at the AIS, as KV1 channels are low threshold, fast activating, such that they act like a brake on action potential firing. So without KV1 channels at the AIS, the neurons fire sooner and they fire more action potentials. And we provide additional evidence in the paper for KV1 channel role in these action potential changes. So these data lead me to our hypothetical model, which is that PSC93 interacts with DHC14 by its PDZ ligand, allowing DHC14 to palmitylate PSC93, binding it to the membrane. This allows PSC93, or may allow PSC93 to interact with KV1 channels, thereby allowing DHC14 to palmitylate the channel. So we were really interested and very excited by these findings, as they have broad implications for understanding ion channel physiology, and as well as understanding disorders characterized by hyperexcitability, such as epilepsy. And so we are following up on these findings in my own lab now. So I'd like to quickly thank Gareth Thomas, my postdoc supervisor, as well as Sally and Santi, and other members of the Thomas lab, and Tasso Zagunas and Hunso, who did the electrophysiological work, as well as our funding agencies. And finally, my own lab, who are working very hard to get experiments going and get their lab up and running. And I look forward to one day telling you about their work in the future. So thank you everybody for joining us today and for your attention.
Michelle, that was great. That's a lovely story, isn't it? So carefully worked out as it worked along. I'm sure there'll be lots of questions on that. Look forward to reading those. But we'll crack on. And our third invited speaker is Michelle Sahai, who's senior lecturer at the University of Roehampton. Her research focuses on understanding the functional mechanisms of membrane proteins and their interactions with cellular components that regulate their function. She uses computational methods of atomistic modeling and will describe her work investigating interactions between new psychoactive substances, formerly known as legal highs, and the dopamine transporter. And I apologize, I think I called Shaun Michelle when I posed her talk, so apologies for that. This is Michelle, previous talk Shaun. Over to you, Michelle.
Thank you very much.
Your slide,
yes, okay.
when your slides are up and running.
Yeah, can you see that?
There they are.
Okay, thank you. So I'd just like to thank the Biochemical Society in the first instance for giving me this opportunity to present my work. So I did give a title called Elucidating the Molecular Mechanism of Action of Stimulant New Psychoactive Substances that Target the High-Affinity Transporter for Dopamine. But I think it's a bit of a mouthful, so let's simplify that a little bit and really go to the crux of it, using computers to study addiction and the brain. So I know based on our audience, I think a significant number of you are actually students and early career scientists, and especially for those of you starting their journey, I wanted to briefly show you my own academic journey. I know I'm a bit privileged to have a traditional trajectory where I spent my undergrad and my master's at the University of Toronto, and then I moved over to Oxford to do my PhD and to New York to do my postdoc at Weill Cornell, and then now back to London at the University of Roehampton, where I'm a senior lecturer. I'm also going to add in a little variety, and I'll sprinkle in some flavors of paprika and vino when I say that I also spent a year abroad between my BSc and my MSc doing research in Szeged and Bologna. And I put this up especially as some advice or inspiration for those of you starting their journey. And I guess caveat being that we're in a pandemic right now. Let's think about what will happen when the world does come back to normal. It's a good idea to not stay in one place. And of course, if you can be comfortable because it's a good place to be and where you feel supported, but try to be as mobile as you can be, because it's always good to meet different people, see different places, and gain a different perspective. And I can tell you that these experiences I have shaped me as a researcher and allowed me to communicate better with my peers. So let's hopefully those lessons that I will learn will help translate to you how I tell you about how computers like this on the left can help you understand addiction and the brain. So we're at a point in time where the convergence and interconnectivity of all these disciplines that you're seeing here can be used by computers to help answer questions in any of these fields, as long as you have the imagination and creativity to ask the right questions. We're going to spend a lot of time in this talk on the periphery of biochemistry and chemistry, but at the crux of it, at the foundations, we're using physics and mathematics and computer science to answer those pressing biological questions. And where all these fields really converge is with the science of molecular computations. And I say this because it's a science that elucidates and validates experimental evidence. So you're using visualization and rationalization, and of course thinking a little bit about putting in some imagination. Of course, you're not really going to have this rainbow striped zebra. But what you want to go down to really is to understand the genotype and down to the atoms and molecules that decide the phenotype that you're looking at. And of course, this is something that we see, and you've seen in the previous two talks, looking at a total whole system approach and coming down to the reductionist approach. And of course, this method is applied in many areas of research, both academic and industrial. Now, structure-based drug design is really at the center of this, and next to this is the fundamental key is the lock and key analogy, where we ask crucial questions like which structure of the lock should be targeted, which key fits best, and is the binding pocket a good target. So our lock in my story is the dopamine transporter, which is a member of the sodium symporter family. And its action is at the synaptic junction between two neuronal cells. And you've seen this before in some of the previous talks. So once neurotransmitters are released at the synapse, they go on to elicit an impulse by binding to receptors in the postsynaptic terminal. And any leftovers are recycled by the dopamine transporters and the other NSS. Functionally, they're important for cognitive, emotional, and behavioral functions, and any sort of dysfunction leads to neurological diseases. And in that being, they also targets for antidepressants, but also drugs of abuse, which is what this talk is going to be about. In the bottom picture, you'll see that specifically coming from, this was a picture from NIDA, where cocaine, it actually blocks the reuptake and the recycling of the neurotransmitter. And as a result, there's an intensity of the stimuli. Over time, these cells become sort of desensitized and get used to the reinforced, rewarding, and pleasurable effects that you get from these drugs like cocaine. So I guess the take-home message here is dopamine is our dopamine transporter is our lock, and it is an important therapeutic target as well as a target for drugs of abuse. We always like to talk about structure and function, and for us, structurally, the information that we get about the three-dimensional structure for this protein comes from site-directed mutagenesis and cross-linking experiments. It tells us about the general fold, the number of helices, and the placement of the terminal ends. But I'll just put in there as a disclaimer, there is no crystal structure for the human dopamine transporter, but that's not going to stop us as computational chemists. So that's our lock, and here is our key. Our key is the substrates or the drugs of abuse. Of course, there's a neurotransmitter, and there are typical drugs like these amphetamine and cocaine that you've seen. But what about these novel psychoactive substances that we're seeing? So if you're not aware of them, they're usually referred to, well, they were referred to as legal highs or marketed as not for human consumption, bath salts in the States, room notarizers. What's really important about this is that they're highly addictive and there's an unknown clinical toxicity. There is the idea that maybe it's an emerging or established because the United Nations Office on Drugs and Crime is still constantly reviewing this list and finding new emerging drugs in different markets and also in vulnerable populations and countries. These are some of the past headlines, you know, because you see this, even though there's been a blanket ban in 2016, you still see this being used because of the clandestine chemists that are coming out with new compounds on a regular basis. And especially because of the pandemic, people locked up in their houses and not finding anything constructive to do, or maybe they're just being bored, they want to try these drugs. So now all these drugs are not the same. They can be categorized simply by the drug wheel because psychoactive drugs affect different people in different ways, and some drugs fit into a number of different categories. So as an example, the cathinones can be having both stimulant and empathogenic effects because they're associated with altered levels of the neurotransmitter levels. So how do researchers identify these popular NPS currently? So you can either find it from the hospitals, the jails, what people are using on the street, or amnesty boxes, where at the end of a night at a party, you end up leaving whatever's left in your pocket in these boxes. And we usually see two very common compounds coming out: the cannabinoids that target the CB1 receptor, and the stimulants and empathogens that target GPCRs and the neurotransmitter transporters. There are also opioids, as you would know, but these are the most common ones. And the one that I'm going to be looking at is the stimulants. And these are classified, I guess, as cocaine-like. Now, this is not Pablo Escobar's cocaine or Walter White's meth. So anyone who's watched Narcos religiously like me would know that Escobar was proud of his product being the purest. Another TV show, all the fictional, was Breaking Bad, which showcased the chemist Walter White and his very blue methamphetamine. Now, clandestine chemists in underground labs are not only using inferior agents in the manufacturing process, but also toxic and hazardous materials that pose a risk to your health and also the environment. So how do we study the effects of these drugs that people are taking, not understanding the toxicity? And these are the neurobiological techniques. So I'll begin with one of my colleagues, Yolanta Opaka-Joffrey at Roehampton, and she uses autoradiography to do competition binding experiments to see that when you soak rodent brain slices, a radioactive cocaine analog, it shows up in red. And then when you bind in a concentration dependent manner, the novel psychoactive substance, example 5-APB here, you see that the drug displaces the analog. So take-home message is really that the drug binds at the dopamine transporter and it can displace cocaine. Another experiment that we've conducted with a collaborator, Colin Davidson, is looking at fast cyclic voltammetry, where you're looking at electrodes and changing the voltage and looking at the current. And what you're seeing in these cells is there is an increase in basal dopamine that blocks the reuptake of dopamine. So what that is consistent with is that this compound is more similar to amphetamine than cocaine. So when I joined, I looked at 5-MAP-B, and this is really pretty much the same. We did the same sort of concentration-dependent binding. We also looked at the experiments with fast cyclic voltammetry, and eventually we started looking at bindings, looking at the structural data. And once you look at the dopamine transporter itself, we wanted to get a structure for that, for our lock. And to do this, because we did not have a crystal structure, we found the template of the Drosophila fruit fly as the best template to make a homology model. And of course, with number of iterative steps, we were able to get that homology model and create this three-dimensional structure, and then dock our compound 5-MAP-B. And what you end up seeing is that there's a comparative a binding site for all of these compounds. Now, of course, finding the spot where these things bind is one thing, but the protein itself lives, and there's a conformational change. So we look at an alternating access mechanism that looks at the series of steps in the transition pathway of the substrate. And if you remember back to this diagram that I showed you initially, cocaine itself blocks it in this particular conformation, which is the outward facing, and amphetamine in this inward facing conformation. So we wanted to see if we could access these different states using our molecular computations and looking at the conformational change and looking at very minute things like these helices that you would see. To do that, we basically model and our compounds and on our protein, embedded in a membrane, think about biologically relevant systems having water and salts, and then end up having long simulations where we can actually look. And if you take a look at this red helix there, you can actually see, I'll just reset it, you can see it starting to move over the course time of this trajectory, and a sodium molecule is going to start popping out of that. And from this experiment, we can actually see the transport mechanism. And with that, you can trace how sodium can move out of the binding site and different interactions, such as residues that may come out from the binding site. So of course, the NPS that we're looking at are very structurally diverse. They're not just amphetamine-like like the ones I just showed you. So we need to consider that, as well as the fact that most of the functional studies are being done on mouse and rat. So we need to also look at the homology modeling of the mouse and the rat dopamine transporter. And we eventually started looking at different compounds, such as these ones here, which are functional groups and the placement and more efficient binding, looking at the functional groups at ortho, para, and meta, and using free energy calculations, we were able to compare those to IC50 values. And additionally, we can look at different compounds and see not only amphetamine type binding, but a compound called 2-DPMP, which is our latest one. Looking at the same sort of binding experiments and modeling, we were able to see a profile that binds more like cocaine than amphetamine. So in future directions, we're actually going to be looking at the other NSS transporters and seeing if we can use these now established methods to see if we can see the same binding in SERT and NET, and with different and other classes of drugs, such as ones that may bind to both, not just specific ones, and also maybe ones that bind to all 3 of the transporters. And with that, I'd like to say thank you. And of course, because of most of this work started off while I was a postdoc at Weill Cornell, I'd like to thank my supervisor and George, and obviously everyone that I've now moved over to the University of Roehampton, and all of the granting bodies that helped me with the computational time and of course the money. So thank you very much for that.
That's great. Thank you, Michelle. I found that particularly interesting because being psychopharmacologist, that's very close to my own field. I've always worked on catecholamines, so I found that fascinating. Thank you.
Thank you.
And now we move on to our final speaker today, who's Dr. János Haller, who's an assistant professor in biomedical sciences at Dublin City University in Ireland. Hello, János. His research uses super resolution microscopy techniques to investigate astrocyte dysfunction in neurological disorders and as a target for novel therapeutic interactions. János is going to explain how using this technique can help to reveal information about the relationship between astrocytic processes and synapses across the brain. Over to you, János. And I'll flag, yes, we're in business. See you later.
Great. Thanks, Claire, for the kind introduction. And thank you very much for the Biochemical Society for giving me the opportunity to present this work to you today. And as Claire said, I'm an assistant prof in GCU, but the work I'm presenting today was done at the Queen Square Institute of Neurology at UCL in London, where I was a postdoc there. So yeah, we've heard a lot about neurons today, and most people always forget that there's another class of cells in the brain called glia as well, that is equally as important, I would say. So I'm particularly interested in a cell type called astrocytes. They're called astrocytes due to their shape, meaning glia, star-shaped cells, sorry. And this is a beautiful drawing by Cajal here. And you can see the cell body and then these main processes that are radiating throughout the brain. And astrocytes tile the entire brain, and they're very important. They have a lot of very important functions. For example, they're involved in waste clearing in the brain. They transport nutrients from the blood vessels to neurons. They also have a very sort of a classical function of these glia cells, the glue of the brain, the connective tissue. So they're holding neurons and other cells in place. They're also phagocytic, so they digest dead parts of neurons, and they're involved in synapse formation during development. And they also regulate the content of the extracellular space. So we've heard in the previous talks but a lot about synapses and the release of neurotransmitters and the reuptake of neurotransmitters. And one of the neurotransmitters, glutamate, is taken up by astrocytes, for example, and then recycled and given back to neurons. And they're also involved in potassium buffering. And when these functions go away, this can lead to hyperexcitability and then seizure generation and diseases such as epilepsy. So synapses are made up of a pre and a post synapse, synaptic part, but synapses in the brain usually are sheeted by these astrocytic processes. And this is then called a tripartite synapse. So you have the pre, the post, and the astrocyte. And in this image here, all images here are series section 3D EM reconstructions. And in azure here, you see the astrocyte, and then all the other colors are dendrites and spines. And what you can see here is that the astrocytes are sheeting or growing around the synapses. They're sheeting mushroom spines as well as thin spines. And that's very important for the function of the synapses, kind of stabilization, but also this reuptake of ions and neurotransmitters. So these processes are extremely small, and synapses themselves are about a micron in size, but these leaf-like structures from the astrocytes are very, very tiny, and their size is below the so-called diffraction limit of light. So this was a theoretical limit proposed by Ernst Abbe, I don't know, 100-something years ago, that whenever you look at a point source of light through an optical system, it will always be blurred up, and you see these kind of iridescent patterns here. And if you look at a GFP molecule, which is only 2 by 4 nanometers, so it's teeny, teeny, tiny, if you look at it, you actually see a big blob of about 400 nanometers in size. So nothing below this diffraction limit can be visualized as a teeny tiny point. So the diffraction limit is about 200 nanometers. So that's fine if you have, if you're only looking at one molecule or your molecules are really spaced out. But as we all know, in a biological system, it's extremely crowded. The cell is just filled with proteins and all have specific functions and they do stuff. So when we do fluorescent microscopy, we just see this haze, this blur of your image. So here in this example here, when your molecules are closer together than the refraction limit, you can't visualize them. You don't know whether it's one molecule, two molecules, three molecules, and so on. So over the past couple of decades, a lot of methodological advances have been made to break this diffraction limit. And most of them are called super resolution microscopy. And the technique that I'm using is called single molecule localization microscopy. And this relies on the sequential blinking of fluorophores. And then you can fit a Gaussian distribution over this, pinpoint the center point of the fluorescent blinking event, and then you can reconstruct the final image, let's say. And just like the Eiffel Tower here and all those light bulbs on the outside of it, you just film the blinking of them, and then you can reconstruct the structure of the Eiffel Tower. And this has been done, for microtubules here in this example and synapses on the bottom. So I've been using this technique to look at these tripartite synapses. So I used bassoon as a presynaptic marker, HOMA1 as a postsynaptic marker, and then looked at glutamate transporter on astrocytes here. So first of all, I did this in vitro. So in cell culture, I cultured mixed hippocampal cultures, so neurons and glia cells together. And again, I stained for bassoon, HOMA1, and GLT1, and I did 3-color 3D SMLM. So this on the bottom here is the fully reconstructed super resolved image. And if you zoom into one of these spots here, it's flying into view now. You see the synapses here in green and magenta, and then in blue you have your glutamate transporter. So in contrast to kind of your average normal epifluorescent image, you can actually visualize bassoon and HOMA as two distinct clusters. So the synaptic cleft is in between them, and the cluster is actually about 160 nanometers apart from each other. So you don't just see sort of a white blob here. But what you can also see is that GLT1 is sort of on the bottom of the synapses. So these three images here on the bottom are color-coded by depth. So the darker the color, the closer the molecules are to the cover slip. So the astrocytes in culture are basically growing on top of the cover slip, and then you have neurons growing on top of them and forming synapses and so on. So unfortunately, in vitro, you don't really get tripartite synapses. So the astrocytes aren't really engulfing synapses, at least in this example here. So we had to move on into situ or into brain sections. So again, I stained for the three molecules, bassoon, HOMA, and GLT1, and then super resolved them. And again, what you can see here in green and red now are the synaptic clefts, so the two clusters of bassoon and HOMA1 on the bottom. And then you see this cloud of GLT1 that's really surrounding the synapses in all kind of dimensions, or 3 dimensions anyway. And with this technique, then you get all these individual localizations, which is visualized here. So each individual dot here is one individual molecule or one blinking event of the fluorophore at least. So then you can count the distances between all these different localizations, and you can plot them. So here, this is the distance between HOMA1 and bassoon, and they're clustered at about 140, 160 nanometers, which is kind of the distance between the pre and the posts. Between those molecules within the synapse. And then you can also plot the distance of the GLT1 molecules to the synaptic compartments. So this is basically just the same again. These are epifluorescent images here of GLT1, bassoon, and HOMA. And then these are the super resolved images here. And you see nice kind of synaptic clefts and these cloud of GLT1 molecules, as you can see here. And this image is just to illustrate sort of the power of super resolution microscopy, or the difference between the two resolutions, let's say. So when you have green HOMA1 and red bassoon, and classically they would co-localize, they form something yellow, which means, oh, they're interacting with each other, they're close by, where in fact, as I said before, there's about 150 nanometers between them. And you can really visualize nicely the synaptic cleft technique. And then you have the cloud of GLT1 molecules here. So I did this some time ago now, and we are kind of finalizing this. And we really wanted to not just make pretty pictures, but we wanted to do a little bit of analysis and get some information out of these images. So we wanted to visualize this 3D tripartite synapse environment in a 2D fashion. So these images on the top here, you have your x-axis and your y-axis, and then Z is collapsed onto the y-axis, and the graph on the right here is just going along x-axis and then collapsing y. So you have your two peaks here for Homer and bassoon, and then the blue line is the distribution of glutamate transporter molecules. And you see that there's sort of more glutamate transporter on the postsynaptic side, which has also been shown in the EM images before. And then this on the bottom here, or in the middle, is the radial distance. So rather than just collapsing the y-axis here, you start at this point and then you go around 360 degrees and you're collapsing everything as you go. So this was a good way of visualizing the distance between the pre and the post synapse. And these peaks are about 160 nanometers apart from each other. And then we rotated the whole image so that the pre-synapse is on the top and the post-synapse on the bottom, which is usually the way that synapses are displayed. And then again, we looked at the radial distance and the distribution of GLT1 molecules surrounding the synapses. And you can see that there's a peak of about 200 nanometers or so away from the actual synapse where you have glutamate transporter molecules. And this kind of makes sense if you imagine that there's a synaptic bouton, also the pre-synapse, and then you have your spine of the post-synapse, which is kind of in the way, and then these perisynaptic associative processes are sort of invading the synaptic cleft where you have your GLT1 molecules. So these images were all done in naive cells or naive animals, in kind of wild type animals. But we also wanted to look at what happens to these processes in plasticity. So this was a collaboration with lots of different people and labs around the world now. The induction of long-term potentiation, so the underlying mechanism of memory formation in the brain. And we wanted to look at what happens to these associative processes during the induction of LTP. And if they retract from a synapse, this can lead to spillover of neurotransmitters. So the glutamate that's released at this synapse here can then escape the synapse and actually activate synapses that are nearby. So associative processes are there, and kind of they are a spherical hindrance, so the glutamate molecules are sort of bouncing off them and they can't escape the synaptic microenvironment, and then they are also taken up by glutamate transporters. So I played a sort of a minor role in this paper, but we looked at this with different techniques, and I kind of supplied these 3D SMLM images. And what we found was that after LTP induction, you have a retraction of associative processes, and you have a sort of a retraction of the glutamate transporters as well. So there's less glutamate transporters close to the presynapse than there is in under control conditions. And this is it. So thank you very much for your attention. And this work was done at the Institute of Neurology in ECR in Professor Dmitry Ruzikov's lab, and it was funded by the ERC and the Wellcome Trust. So thank you very much.
Gosh, thank you. That's just amazing. See those images. I didn't know about that technique, so I've learned a lot. Thank you very much for that. We are running over time a bit, but I think we've got time for at least one question for each speaker. So if we can just run through this. And of course, you can follow up on these questions online later on if you want to. So it's not a now or never opportunity. So one for Lewis. How many of the proteins that were upregulated were astrocyte proteins? And do you think it might reflect changes in the astrocyte neuron ratio rather than changes in expression per se?
Sorry, I missed the first point because I
okay.
was just being unmuted. Sorry.
Oh, okay. Sorry. It's a question for Liz. Many of the proteins that were upregulated were astrocyte proteins, do you think that this might reflect changes in the astrocyte neuron ratio rather than changes in expression per se?
Well, thank you for that. There were many other proteins that were upregulated. I mean, we found almost 5,000 proteins, but in terms of significant upregulation, we are talking about almost 400 proteins. And of course, some of the key proteins that were upregulated were the astrocyte proteins like GFAP and vimentin, and that could reflect astrogliosis and neuroinflammation, which is a response to the neurons being infected with a prion, and also a response to the immune system as a whole. So yes, we can show from the data that there is an increase in astrogliosis, which is characterized by, of course, the significant upregulation of GFAP and other proteins related to the astrocytes.
Right, thank you. That question was from Philip Kitchen, by the way. I don't know if you know him. I'll move on to a question to Shaun. Shaun, this is from Sophie Bradley. Thanks for a really nice talk. Are there any known links between mutations in ZDHHC14 and human epileptic patients?
Yeah, that's a really great question, and I would love to find one or some. But in all the kind of databases and data sets that I've looked in, no. What we do see is using the, I think it's the genome or nomad, or anyway, one of the databases, if they look at loss of function mutations, DHC14 is resistant. It seems to loss of function mutations in humans, which kind of suggests that it's an important gene. But yeah, no, you know, mutations or variants that we know of yet that that might in DHC14 that might lead to any kind of human disorder. We are looking at some mutations that occur in the KV1 channel around the palmitoyl site in human mutations that lead to episodic ataxias and seizure disorders. And so we're really interested in knowing if any of those human mutations cause or changes in palmitylation and AIS targeting.
Thank you. They're very concise answers. That must have been very impressive. Thank you. One for Michelle. I have one lined up, sorry. I want to try and make sure we go. Well, while I'm trying to find, I have one ready for Michelle. But Janosch, here's one for you. Did you ever try expansion microscopy for your synapse experiments?
Sorry, I missed that. Was the question whether expansion microscopy can be used?
For your synapse experiments, from Jonathan Warboys.
Okay, yeah, no, I mean, expansion microscopy, yes, has been used kind of for this for a while, I suppose. Yeah, there's beautiful kind of images that really work. So expansion microscopy, you just expand the whole tissue to kind of increase the distances between the individual molecules, and then you can localize them or visualize them as well. So yeah, I've tried expansion microscopy as well in the past, and it definitely works. Yeah. I don't know if that answers the question, but yeah.
Great, thank you. And Michelle, from Sophie Bradley, really interesting talk. Thank you. Two questions actually, if you can answer them both briefly. Are you or others attempting to crystallize the dopamine transporter? And also, do you test NPS at a panel of other targets besides DAT for binding or activity?
Okay, yeah. So I'm not a crystallographer, but there are other labs that do the crystallography. So as I mentioned in the talk, there is a crystal structure for the Drosophila, so the fruit fly, dopamine transporter. There is a crystal structure for even the human serotonin transporter, but there is none for the dopamine transporter. And I'm assuming that those people, those labs who are versed in X-ray crystallography are working very hard to get it to us. So fingers crossed. And the other question is, are we looking at a panel of different targets? At the moment, within my own research, no. But I can also tell you that there is a limit to how you can do this, because these compounds, of course, they're illicit. And once they appear on the market, or the UNODC sees them, their main the importance of what they're doing really is to do some experiments to identify what it is. They don't really spend too much time on accessing different targets until it gets filtered down to people like me. So of course that's going to end up with the researchers who are doing the functional assays. So some people are doing that, but not particularly me. But if you're interested in the labs that are doing that, I can obviously point you in the right direction. But because these are illicit compounds, there's a lot of licensing and agreements that have to come about, because of course we don't want this information getting out to the wrong people who might want to make these drugs bind better, if I can put it that way, right? We don't want them to make them more potent. So of course the information is yes, protected exactly, very closely.
Thank you. Good answer.
Thank you.
Yeah. Question for Lewis from Neve Doherty, who I guess is Irish, I think. The abnormalities related to Alzheimer's, which are reversed by the treatment, would these be reversible in more advanced stages of the disease in mice, or would you expect to see a reduction in speed of disease advancement?
Yeah, that's a very good question. So as people who work on drug development, we have on the different ways that we can target Alzheimer's disease at different stages of the disease. So with the positive allosteric modulator I mentioned today, you want to upregulate the ACH transmission, but this will only happen if you have some amount of endogenous acetylcholine present. And so this kind of situation should happen in the early to mid stages of the disease. But when you get to the more severe stages where your ACH acetylcholine levels are almost gone, you should be thinking about using orthosteric ligands, or even using positive allosteric modulators that can also bind to the orthosteric pocket. But you need to weigh all these against the side effects and the risk factors that come with it. So we have different ideas of targeting different stages of the disease with different drugs.
Okay, thank you. Question for Janos from Philip Kitchen. You mentioned that you can't recapitulate tripartite synapses in 2D co-cultures. Have you tried 3D or organ-on-a-chip culture systems?
Yeah, thank you very much for the question. It's a very good question. I have not really tried it. I tried kind of staining synapses nicely in organoids with a collaborator, but we didn't really get all that far down the line. At the moment in my lab, we don't have any kind of organoids and any 3D culture. So one of the problems with astrocytes is that when you culture them, they really change their gene expression profile. So they aren't really, the same cells as they are in situ. And that's again one of the reasons why the nice 2D culture doesn't really work all that well in studying astrocytes. So that's why it's a good suggestion, to use kind of 3D culture models in mAChR or something like this. I haven't tried it, but it's definitely, I suppose, an easy way, and you get nice images there as well. And I should definitely try it.
Okay, thank you. And I got a final question for Shaun. Do you expect this system only to, I don't know about the distribution of those potassium channels, do they occur only in the hippocampus, or would you expect it to generalize to other brain regions?
Yeah, that's a really good question. Yeah, and certainly they aren't only expressed in the hippocampus, so we would expect to see kind of similar things in other brain regions. DHC14 is very, very highly expressed in the cerebellum, so that's something we're really interested in following up on, as well as PSD93 in the cerebellum. But yeah, I would expect to see kind of similar things in other brain regions.
Right. You've got to start somewhere. So
yeah,
the hippocampus is a good place.
Yeah, they're easy neurons to grow.
Well, look, we have got loads of questions here. So if the speakers, would be kind enough to check through those questions, and if you can interact with the questioners sort of offline, that would be really good. And you can see there's been an enormous amount of interest in all your talks, and I think you'll find many of those questions really interesting. So do that. But to stick to time, I'd probably better wrap up now. And obviously, I want to thank everybody for attending the webinar, and in particular to the 4 speakers for volunteering to contribute to the webinar and for giving those 4 truly fantastic talks. And I learned an awful lot this afternoon. Really fascinating, impressive stuff. And as I said, you can carry on this conversation online, and you can follow at BiochemSoc via Twitter, and also PPP Publishing on Twitter as well. So watch out for that. And as I said right at the beginning, we're always glad to have suggestions for future topics for webinars and also speakers. And so if you've got an idea for a webinar, do submit that proposal. You can find more information about that and also future planned webinars on the Biochemistry Society website, which is www.biochemistry.org/webinars. So do have a look at that. And if you've missed any of the 20 or more webinars that we've run in the past in 2020, or if you'd like to watch them again, just go to the website or the YouTube channel, and you can get their details on the screen right there. So make a note of those and you can catch up on what's happened in the past. And you can join us in our next webinar, which is on obesity, from genes to biochemistry of a global pandemic. And that's going to be on Thursday, the 28th of January, which is in just over a week's time, at 1400 hours, Greenwich Mean Time. And at that session, you will hear from Nicholas Morton, who's the personal chair of molecular metabolism at the University of Edinburgh. And he will cover discovery approaches to obesity genes using mouse models and a GWAS approach, and also the gene biochemistry, proteomics, and so forth. The webinar will be chaired by Professor Roger Cox, who leads the genetics of type 2 diabetes group in the mammalian genetics unit at the MRC Harwell Institute in Oxfordshire. So these are very challenging times, I hardly need saying, and it's even more important than ever for us all to stay connected. So it's an exciting time to join the Biochemistry Society, because they're looking to expand all these online activities that are helping us all to connect with each other. And if you join that community of researchers and specialists, it will help you not only stay connected, but it will also help you take advantage of all the benefits which the Biochem Society offers. And they include discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, and personal online access to 2 of the journals in the Portland Press portfolio. And there are more advantages than that, but I don't want to just give you a very long list. But do visit the Biochemistry Society website and find out much more. As I said, we want to do all we can, particularly to encourage early career researchers. So take advantage of that. So at the end of this fantastic webinar, I was pretty stressed out about taking this on because, you know, it's not like doing an in vivo one. All sorts of things could go wrong, but as far as I'm aware, nothing's gone wrong. So it's a great tribute to the team behind the screen who are organising all the buttons. You've no idea how much effort they've put in to organising this, and it's run so smoothly. So thank you to them, and thank you to the speakers once again. And I hope we all meet up again in vivo one day. Thank you. Bye-bye.